All right. I think we'll get started. There might be a few other folks trickling in, but I want to welcome everybody. I'm Tova Burstein, Community Engagement Director with the FDMAS Alliance, and I'm really pleased to be here with you for the 2025 Team FDMAS Research Update. Few quick reminders before we get started. We are recording today's program so that we can share it later with the wider audience and community. So please keep your microphone muted unless you're presenting. The chat is always open. It's a wonderful way to include questions and discussion while we're going. We have two presentations happening today, and there will be time for questions and discussion for those presentations. And you can also, again, send the questions in via the chat. We're thrilled to have you here. And for those who are unfamiliar with our organization, FDMAS Alliance is a community-driven nonprofit devoted to the development of evidence-based treatments for fibrous dysplasia McCune-Albright syndrome, or FDMAS. We advance research, provide education, and channel the voices of those affected by FDMAS. And all of our work is made possible by your donations. So thank you so much to all of those who have supported this work. All of the work that you're going to be hearing about today is made possible through donations. And again, we're very grateful to have you here. With that, I'm going to pass it over to the Executive Director of the organization, Adrienne McBride, to say some quick thanks.
Thank you, Tova, and thank you for all the work that you've done to make this webinar possible. Before we move on, I'd like to take a moment to briefly recognize some of the incredible individuals and groups who have played a key role in the success of the $1 Million Bike Ride. First, a huge thank you to our previous $1 Million Board MDBR awardees, Dr. Fernando Fierro, Dr. Julia Charles, and Dr. Biagio Palmisano. I know I'm butchering that name. Is there anybody else who I've missed? Because we're really proud of everything that you've done. I also want to recognize our friends at MASFD, specifically Lily Dagno, for her partnership and the work and her contributions. And I also want to acknowledge our FDMAS Alliance board directors who are joining us today. I see Ann Corvell and Nikki Dealey. Is there anyone else? And now I have the privilege of turning the presentation over to Cindi Brandt Levin. Cindi is the FDMAS Alliance Vice President and the unstoppable force behind Team FDMAS in the UPenn Orphan Disease Center's $1 Million Bike Ride. She's been championing this initiative since 2014, and her commitment has been truly transformative. Beyond the $1 Million Bike Ride, Cindi devotes an incredible amount of time, energy, and heart to our organization and to the FDMAS community. Her leadership and her determination continue to move our mission forward in powerful ways, and we're so grateful for everything that she does. With that, I'll turn it over to Cindi. Cindi?
Are you there, Cindi? We can't hear you. Do we lose her? She's here.
Can you hear me?
Yes.
Now we can.
Now you can hear me?
Yes.
Yes.
Oh, okay, great. Sorry about that. Thank you for that introduction. And I thank everyone who Adrienne and Tova have thanked as well. It is a privilege to be here with people in the community who have been supportive of Team FDMAS, and most importantly, the researchers here who, without your devotion to our cause, we would not be where we are today and made the types of strides that never in my 11 years working with this organization would I ever imagine we would be where we are today. Thank you, Adrienne and Tova, for organizing this great webinar as well to update us on the research. As the slide you're viewing right now has summarized, we have raised $1.2 million in direct research grants through the $1 Million Bike Ride at UPenn. And through these pilot studies, we have secured numerous collaborations and have an ongoing $6 million further research funding through the Department of Defense. This year, is especially important that we rally for Team FDMAS, as we all know what is going on in the current climate with losses in federal funding and other research in jeopardy. This Team FDMAS fundraiser that we're having this June 14th is probably the most critical fundraiser I have had in all of my 11 years in this organization, doing the $1 Million Bike Ride. So anyone who can support us during this time, please donate, ride, spread the word, because we need to support the FDMAS Alliance and all the work that we are doing in terms of advocacy on the Hill, in terms of keeping all of the research going and the labs not getting closed, and everything else that our entire staff and organization is devoted to is part of this Team FDMAS initiative, and especially this year. So I hope to see you all there, or see you join us in our fundraising for this great cause. And I will turn it over now to Tova to introduce our esteemed new colleague on our board of directors. Thank you.
That's right. Thank you, Cindi. So yes, I have the privilege of introducing somebody who I think needs no introduction. Dr. Collins is recently retired, although still very active. Mike, are you stateside now, or are you still traveling?
No, I'm here.
Oh, you're here. Okay, you're here. Recently retired from the Skeletal Disorders and Mineral Homeostasis section of the NIDCR at the Natural History Study with FDMAS. And he's devoted his career to the study of fibrous dysplasia McCune-Albright syndrome, including close collaboration with this organization since our founding. You served as the medical advisory chair for over a decade, and you're currently the scientific advisory chair, and recently, as Cindi shared, a member of the board of directors. Mike, we're so grateful for you, and I will turn it over to you to lead our discussion.
Thank you for the introduction, Tova. And this is one of the favorite things I like to be involved in and see is the progress in the research that's made, because it really is, I think this is textbook what an organization like FDMAS should be doing to give seed grant funding. This grant funding is turned into larger grants, and the research that we're going to see today is a big part of that and really impressive. I also want to thank Cindi for all she does with the $1 Million Bike Ride. It's amazing, the patients, the families. And I won't take any more time for that, but I will emphasize that this is really a critical time in funding from the U.S. government, and anything anyone can do to, and I can say this now because I'm no longer a government employee, anything can do to speak to their elected representatives to emphasize to them how important this research is to all of us here in the States and around the world, as you'll see from our speakers as well. I think that's really important. Okay, so that's enough. So I'll introduce, and I think the decision was Mara is going to go first. So Mara and I have known each other for a very long time. We've worked together for a very long time. She has made, and her colleagues have made seminal contributions to our understanding of FDMAS. They created the first animal models for this. They've done amazing work with it. And this work continues to this day. And it's great that the $1 Million Bike Ride is been able to support this. So with that, I look forward to hearing Mara's update.
Okay. Thank you, Mike, for this very nice sorry. Okay. Okay. Can you see my screen? Okay. Okay, so thank you, Mike, for this very nice introduction. Thanks again for giving us the opportunity to show our work to the FDMAS community today. This is our 2023 project, which was based on part of our previous work, supported by multiple MDBR grants, and performed on this mouse model of fibrous dysplasia. And the project had actually, it had multiple aims. I'm going to focus on these two aims. The first one was to attempt to identify a strategy, specifically a drug combination, able to maintain the effect of the anti-rank ligand antibody, denosumab, in humans after treatment discontinuation. Whereas in the second aim, we started to investigate the morphological mechanisms underlying the functional impairment and even chronic bone pain associated with this disease. So the first aim raised by the evidence provided by our and other groups through experimental and clinical studies showing that the anti-rank ligand antibody has a beneficial effect, actually a therapeutic effect in fibrous dysplasia, since by inhibiting generation of osteoclasts, meaning those cells that destroy bones and are abnormally increased within FD lesions, it allows the deposition, the conversion actually of the fibro-osseous tissue of FD lesions into bone, and it also prevents the progression of the disease. However, it's well known that this effect is unfortunately reversible, and after treatment discontinuation, when osteoclast formation starts again and bone resorption, bone disruption is resumed, FD lesions recur, and in some cases there is also an abnormal increase in serum calcium levels, which may be very dangerous for patients. And so in this project, we first asked whether zoledronic acid, which is a bisphosphonate, and as such it does not inhibit osteoclast formation, but it reduces osteoclast activity, and it also persists for a long time in our skeleton because it binds to the mineralized matrix. So we asked whether zoledronic acid could be of help to maintain the effect of the anti-rank ligand antibody, actually to delay the reappearance of FD lesions after discontinuation of the anti-rank ligand antibody. And to address this question, we generated multiple experimental mouse groups, each of which received a different treatment schedule. We were particularly interested in comparing these two groups. The first group was the short, we call it short anti-rank ligand group, because these mice received 4 doses of the anti-rank mouse rank ligand antibody in the first 3 weeks. The second group, we call it the combined treatment, because these mice received the same 4 doses of the anti-rank ligand antibody, plus 2 doses of zoledronic acid, 1 during and 1 at the end of the treatment. And then we also generate additional control groups, the long anti-rank ligand group, in which the treatment with the anti-rank ligand antibody was continued, 4 doses in 3 weeks, and then 1 dose every 3 weeks. Sorry. The zoledronic acid group, which received only the 2 doses of zoledronic acid, and the vehicle group, which did not receive any drugs. And we carried on these experiments for 12 weeks, and every 4 weeks we performed the radiographic evaluation, and we also collected mouse blood. And here are the results. At 4 weeks, meaning 4 weeks after the beginning of the treatment, all mice that received the anti-rank ligand antibody, no matter what the specific treatment schedule was, all of them showed an increased radiographic density of affected skeletal segments, as you can see here, for example, in the short treatment, the combined treatment, and the long treatment. Whereas at this time, we did not observe major changes in the zoledronic acid group. However, at week 12, meaning at the end of the experiments, mice in the short anti-rank ligand group already showed the recurrence of FD lesions. Actually, in these mice, FD lesions recurred as early as week 8, meaning 4 weeks after treatment discontinuation. Whereas at the same time, meaning at the end of the experiments, the increased density, was maintained in mice that received the combined treatment. Obviously, it was maintained also in mice that received the long treatment. And at this time, we also observed an increased density in the zoledronic acid group. These radiographic results were partially supported by histology because in the combined treatment, we observed an increased amount of bone compared to the short rank ligand group. So it was, in terms of bone, it was comparable to the long anti-rank ligand and zoledronic acid. Although we observed some small areas of fibrotic tissue and some osteoclast. And bone turnover markers, the level of bone turnover markers was in agreement with this observation because the levels were lower in the combined group compared to the short rank ligand treatment. So going back to our questions, I can say that yes, at least in this mouse model, the combination of zoledronic acid and anti-rank ligand is useful to maintain, at least at radiographic and bone turnover markers level, the effect of the anti-rank ligand antibody. Although, as I showed you before, there is the reappearance of some fibrotic tissue and osteoclasts. The second aim, again, raised by, was based on some of our previous work, showing that this mouse model may be a useful model to study physical impairment caused by fibrous dysplasia in some patients. Actually, I already showed you these results in my last presentation. Now we have completed the experiment. We have expanded the number of mice that we have analyzed, and we have confirmed that if we test these mice, the behavior of these mice by using well-established tests such as the burrowing test and the nesting test, meaning tests that evaluates the ability of mice to burrow food pellets or to build a nest, we can see that there is a clear impairment in the physical activity of transgenic mice compared to wild-type mice. Whereas in the tail flick test, which measures the ability of mice to, the reaction of mice to an harmful stimuli, we could see a longer latency of reaction compared to wild-type mice. I would like to remark here that these results were in complete agreement with what we observed in human, what has been observed actually in human patients. For example, the long latency of reaction to harmful stimuli has been reported in FD patients. And in none of these tests, as in human patients, we observed the correlation between the results of this test, meaning the severity of physical impairment, and the disease burden, meaning the extension of the disease. And I would also like to remark that this mouse model certainly is not the perfect model of fibrous dysplasia, but it's an important model because in these mice, FD lesions develop spontaneously, progress over time, and generate different patterns of skeletal involvement. So they can be used to evaluate the correlation of any features of any value with the disease burden, meaning with the extension of the disease. And so having assessed that this mouse model can be used to, as I said, evaluate the physical impairment, we asked whether the anti-bone resorption drugs that are commonly used in FD patients may be helpful to improve the functional activity, the physical activity of these mice. And to address these questions, we performed the burrowing test and nesting test on the same experimental groups that I showed you before. And here are the results. If we consider the number of mice that showed a positive change, meaning an improvement in the physical activity, a positive change between the behavior, the physical activity at the end of the treatment compared to the beginning of the treatment, we can see that the number of these mice is higher in all the anti-rank ligand groups compared to the zoledronic acid group and the vehicle group, both in the burrowing and nesting test. And so to answer the question, we can say that at least in this mouse model, it seems that the anti-rank ligand antibody is more efficient in improving physical activity compared to zoledronic acid. Finally, as I said at the beginning, we wanted to start to investigate the morphological basis of this physical impairment and even chronic bone pain in fibrous dysplasia. And we started with the evaluation of the number of distribution of nerve fibers within F deletions. Nerve fibers are those structures that are distributed in all our organs and tissues. They are involved in the perception of harmful stimuli and in the transmission of these stimuli to the central nervous system where pain is elaborated, but you will hear more on this in the next presentation, so I'm not going into a field that it's not my field. We just wanted to perform, as I said, the morphological evaluation, meaning we wanted to evaluate the number and distribution of nerve fibers. And to address these questions in the mouse model, in mice, we actually generated a new mouse model of fibrous dysplasia. It's a triple transgenic mouse model. I'm not going in detail into this from the technical point of view. I just want to say that this mouse model develops F deletions, and in this mouse model, nerve fibers can be easily detected because they are highlighted in green. And so in contrast with what we hypothesize, as you can see here in these large cranial lesions, or here in these femoral and vertebral lesions, the number of nerve fibers within mouse FD lesions is not very high. Actually, nerve fibers are few, and they are mainly distributed around blood vessels as normal nerve fibers do. In this mouse model, all nerve fibers are highlighted in green. And so to confirm the presence of sensitive nerve fibers, which are specifically involved in the transmission of pain, we performed immunohistochemistry demonstrating the presence of this specific subset of nerve structures. And then we wanted to obviously evaluate the same patterns of innervation in the human F deletions. And for this, we analyzed 13 bone biopsies from 7 FDMAS patients. And again, in spite of the large areas of fibrous tissue, in spite of the increased number of blood vessels are the number of blood vessels is usually very, very high in F deletions, in human F deletions. But in spite of this finding, we again could detect only a few nerve fibers in general, in total, and only a few sensitive nerve fibers. So going back to our questions, the answer is that based on these results, it seems that fibrous dysplasia does not cause an abnormal growth of nerve fibers, either in mice or in human samples. Obviously, however, there are many other potential mechanisms that remain to be investigated. For example, if the function of these nerve fibers in the transmission of stimuli is normal, if the elaboration of the transmitted stimuli in the central nervous system is normal, if there is the contribution of other organ and tissues to the unpleasant sensation caused by F deletions. And so this will be probably the subject of future studies. And again, I would like to remark that this mouse model can be very helpful, because in this mouse model that we have used for our experiments, the mutation is expressed in all organ and tissues. So it also expressed in the nervous system, nerve fibers and central nervous systems and other organs. And to conclude, I would like to say that these data are included in 2 manuscripts. The first one is under the second round of revision in Journal of Bone and Mineral Research, whereas the second one is ready to submission. And with this, I thank you for your attention. And obviously, I acknowledge all the components of the Sapienza University team. Thank you.
Thank you, Mara. Tova, do you want to?
Yeah, I think we could do you want to do maybe 5 or so minutes of questions before we then hear from Imran?
Yeah, we have a time. I saw there was one question already in the chat from Derek Miskiewicz. Hello, sorry for the interruption. I have to join another meeting now. So I'm sending my question, which you won't hear the answer to, in advance to hopefully hear the answer during the Q&A. I'll make sure to watch the recording after. It's very good. Thank you very much for your work on this subject, Dr. Mara. It's incredibly valuable and appreciated. I was wondering if the results of your research on fibrous dysplasia in mice are publicly available or accessible for purchase, including how the FD mice were generated and obtained. Mara?
So, Mike, let me understand better. The question
I'm trying to understand it too.
The generation, sorry?
Yeah, I'm trying to understand it. So let me see. If the mice are publicly available, so could he get the mice from you or from a public
Not
repository?
yet. Yeah, not
Okay.
yet. But we can obviously we can give this mouse model to everyone is interested in performing study. Actually, we already did this. We send mice. Yeah, we ship mice to another country for collaboration. So no problem for that.
Yeah, so he should contact you directly with that,
Yes.
I guess, right?
Yes.
Okay, good. All right, so that was that question. Are there any other questions from people who are here at the meeting? I'm trying to, Tova, are there any hands raised or more questions in the chat? Oh, Julia has a question.
Okay.
Mara, I was interested in your thoughts on the zoledronic acid alone treatment group, because at least the x-rays to my naive eye didn't look that different from the long rank ligand, and the histology didn't look that different from the long rank ligand. So do you think that zoledronic acid is actually an effective treatment in your model compared to what I think people have tried? The oral bisphosphonates were not effective in humans, but maybe the IV form is?
I mean, I can tell you that it is effective, but it takes a longer time compared to anti-rank ligand to give an increase in the radiographic density and in the amount of bone. However, you have to keep in mind that with zoledronic acid, it's not possible to completely inhibit bone remodeling. So bone remodeling continues. And my feeling is that if we continue with the administration, we can also get deformities because there is a huge amount of bone that is produced. Okay, so, but it has an effect for sure.
So if no one else has a question, Mara, I have a comment and a question. And the comment is, first of all, that was a wonderful presentation of really wonderful data. You managed to present in a very clear way a lot of research in a short period of time that I think was very accessible to not only the scientists here, but probably to lay people as well. So thank you. That was really great. The one question I had is, have you tried or have you considered a pre-treatment with zoledronic acid before denosumab? And the reason I say that is because we know that bisphosphonates, their effect is best when it's incorporated into the bone matrix. And under the influence of the denosumab-like drug, it's likely that little matrix is being laid down, and maybe you could have a more potent effect if you got the bisphosphonate in ahead of time.
No, I mean, I think that, I mean, we made these discussions before, you know, deciding the schedule, the treatment schedules many times. We know that bisphosphonates binds to the mineralized matrix, to the matrix that is forming and is mineralizing. So we reason that if we give it before denosumab, when the FD bone is actually osteomalacic, maybe there is a little possibility for bisphosphonate to bind, the matrix. Whereas if we give it not only after, but also during the treatment, there are more chances that it can bind to the matrix that is forming. And we know in this mouse model, as we published, that the newly formed bone is also hypermineralized. So we think that maybe the administration during the treatment with the anti-rank ligand antibody could be more effective than before or only after the rank ligand antibody.
Yeah, no, but it's great work. It addresses a real serious issue in the field, and that is, the toxicity related to denosumab use in terms of the rebound, and this appears to address that.
Yeah, I mean, at least I can tell you that we didn't go any farther, but we can gain at least 4 weeks at radiographic and bone turnover marker level. We can maintain the effect for at least 4 weeks. Actually, at the bone marker level, the level is also lower than the long rank ligand treatment or the denosumab alone. So at least for 4 weeks, we have a good results.
Very nice, very nice. So yeah, and Julia, to your question, we know that long-term treatment with bisphosphonates and fibrous dysplasia, those patients are also at risk and develop osteonecrosis of the jaw. So this may allow longer anti-resorptive treatment, this sort of approach.
Yeah, absolutely.
Yeah. Very nice. If there's no other questions, which it doesn't look like there is, are there any, Tova, that you see? I don't see any.
I might have a question.
Go ahead.
Very good.
Yeah. So Mara, thank you. That was a very both congratulations both to you and Biagio and your team, as it's very elegant studies, and I love those green nerves in your mice. You said that your mice would be interesting to us also study the nerves because the mutation is expressed in the nerves. But what about humans? I mean, how does that compare to the human?
Well, I mean, in human, we cannot know because human is a mosaic, a somatic mosaic, right? So the possibility is that there are some patients who have the mutation in the nerves, and there are some patients that do not have it. So I think that having a mouse model in which the mutation is in all the nerve structures compared to a model maybe in which the mutation is not in all the nerve structures could be useful to understand the function of this, the direct, function of this, the nervous system in this disease. I don't know if I answered your question.
Yes, you did. Definitely. Thank you.
Did you hear me?
Someone's speaking, but we can't hear you very well.
Michael White, can you hear me now?
Yeah, you're faint, Michael, but we can hear you.
Okay, better now?
Much better.
Okay. Presumably your mouse model has systemic disease, and then you treat with zoledronic acid and denosumab. But for fibrous dysplasia, let's say polyostotic fibrous dysplasia, in the doses that you're using or in any clinical experience, what might be happening in the normal bones? If you're treating a child, could you be inducing a pharmacologic osteopetrosis in the normal skeleton?
Okay, thank you, Michael, for these questions. First of all, I mean, this mouse model has a ubiquitous expression of the mutation, but the very interesting part is that the lesions do not involve the entire skeleton. I mean, it is a phenotypic mosaic anyway, so we have affected and non-affected skeletal segments. And the dose that we used were comparable, making the right conversion, were comparable to the doses that are used in human patients. We did not observe, at least in these experiments, maybe we should perform longer experiments, but we did not observe particular, adverse effect on the other sides of the skeleton, increase in bone mass, of course, but as it is expected in patients. But it's important to this point that the mutation is everywhere, but FD lesions are still a mosaic, which is very interesting because this implies that there is something else in addition to the mutation that regulates the expression, the development of the disease.
Yes, fascinating. Thank you.
Thank you.
Michael, to your question, clinically, because both growing kids and adults are treated, the approach has been to use the least amount of drug you can to achieve the goal you want to achieve, which in most cases has been pain relief with bisphosphonates and increase in bone density and lesions with denosumab. And with adequate cycling on and off of it, it appears that you can get the beneficial effect without causing a sort of osteopetrotic phenotype. That's the clinical experience so far.
Good, thank you.
Julia has a question.
Oh, it was actually just more a comment to that. In the mouse model that Yingzi Yang developed, when we use anti-rank ligand in the young animals, not cycling it on and off, but using it continuously, you do get a growth plate effect. In short, they don't grow, their longitudinal growth is inhibited and their growth plate is altered. So I think, Dr. White, your concern is valid, and so it just speaks to trying to understand how to best safely use it in kids.
Thank you.
Okay.
Mara, you had a comment?
No, it's fine.
Okay.
I think it's fine.
Okay.
Thank you.
All right, if there's no other questions, and it doesn't look like there are, then we'll move on to Professor Heegaard. Anne-Marie is a professor at the University of Copenhagen in Denmark. She too has been a longtime collaborator and friend, and is not so recently, but relatively recently, joined in the field and research in this area, and has made incredible progress in terms of defining the pathomechanism of pain in patients with the use of animal models. And I think that's what Anne-Marie is going to talk to us about today.
Yes. Do you want me to say something? Okay. I think Mara, I think your phone might not might not be muted. I don't know if that's the anyway. Let me share my presentation. All right. Let me just remove you guys. All right. So I'm going to talk about bone pain and fibrous dysplasia and the work we're doing, specifically the work we've been doing as a result of receiving the grant from the multi-dollar bike ride. And I'd like to start with the acknowledgements. Just a second, I'm just gonna hide this. Yeah. One more second. This work has been performed at my university, and it's been the driving source has been a driving person driving this project has been Chelsea Hopkins, who was doing this under an EU grant for her PhD. And then we had also Julie Bentin, who was hired actually on the grant that we received. And then we have Miguel from Mexico, Jimenez Andrade, who's a professor in Mexico, and his team, who has been doing extensive analysis of the mice that we've been doing, and he was a co-investigator in the grant. We've been very grateful to Mike and Alison, and especially Louis at the National Institute of Health for helping us and providing mice for the study. And we also had AstraZeneca to measure the blood samples from the mice. But I really need to express my gratitude. Let me see. I just want to get, here we go. See if I can get this next one. Yeah. To the University of Pennsylvania Orphan Disease Center and the FDMS Alliance and the $1 million bike ride. I mean, without this grant, we have not been able to bridge from our funding to what we now have, a new project funded by the EU also on pain in FD. And so that's been extremely helpful. So in our group, we work with bone pain, and bone pain is caused by many different diseases in the bone, and we have been focusing on cancer in the bone and also rare bone disorders. And in this case, what I'm going to talk to you about today, obviously, is pain in fibrous dysplasia. And we're working both with mouse models. We expand into the clinic and also looking at mechanisms in vitro. So when we talk about pain, what's important to understand sort of from the clinical perspective is that we have different sort of types of pain. And we talk about nociceptive pain. And this is a pain that you would get which is not damage to the nervous system. So it's actually damage to the non-neural tissue. And it's important because in this kind of pain, one would treat with medications such as, for instance, aspirin. And then we have the neuropathic pain. And this is classified as pain caused by a lesion or disease of the nervous system. So something directly affecting either the peripheral or the central nervous system. And then we have a third category, which I'm not going to address today, which is basically pain where you have an altered nociception. You have you feel a pain, but we don't actually see any tissue damage. So we're not quite sure about the mechanisms. And we can, in the clinical situation, we can ask the patient about their pain, and we can get an impression of the intensity from this questionnaire called Brief Pain Inventory short form, where we ask about the worst pain over the last 24 hours, the least pain, the average pain, the current pain. We ask about the pain medication. And we also ask how the pain interferes with the daily functional. I mean, how does the pain affect the daily activity, the walking, the sleep, and so on. And this has been used with patients for fibrous dysplasia. I mean, Mike Collins' group was some of the first, if not the first, to report on pain in 2008, I think, on pain in fibrous dysplasia. There's also a more recent study from the group in the Netherlands where they used this questionnaire, the Brief Pain Inventory, to get an impression of the pain severity and also the pain interference in patients with both monostotic, polystotic, and McCune-Albright syndrome. And you could see that both were reported from a scale from 1 to 10. There was a pain intensity, sort of an average pain intensity around 3 to 4, and also interfering with the daily functioning. So, but that is just general the pain. But then, which kind of pain? How are the pain? How is the pain classified for the patients? And there has been, one can then use another questionnaire called Pain Detect, which originally was developed to identify like neuropathic pain in neuropathic conditions. I mean, conditions with direct nerve damage, but it's also been used to identify sort of a neuropathic component of pain in diseases such as osteoarthritis and also now in fibrous dysplasia. And in this questionnaire, we ask both about the sensation, the sort of quality of the pain. Is it burning, tingling, prickling? About how it radiates to other regions of the body, and how it's sort of how it's the shape of the pain in a way, does it come constantly or there are pain attacks? And then if depending on the score from this questionnaire, one can either say that a neuropathic component is unlikely or that it is likely or it's uncertain. And one can get an idea of whether there is a neuropathic pain-like component to the pain. And this has been reported both from the NIH and also from the UK. There was community-based registers reported that this presence of neuropathic like pain in fibrous dysplasia. So there was these web-based patient registers, and there was so it's very driven by reporting. So it's in a way that not it's kind of biased in that way, but the results showed that if we look at, you remember the nociceptive pain, which is one that's not from nerve damage, and the neuropathic pain, then we can see at the bottom of this graph to the right, we have the pain intensity, the strongest pain during the last 4 weeks. And you can see the higher the pain intensity is, the more of a neuropathic component is reported by the patients, indicating there might be some affecting sort of damage to the nerves as well, contributing to the pain. And there's another study from Boston, the group in Boston, which also reported that there was a neuropathic pain-like quality to the pain of patients with FDMS. So from the clinical studies, we can conclude that the pain symptoms in FD are probably both neuropathic-like and also nociceptive, which is more sort of an inflammation probably, or other factors in the peripheral. And we still don't really know much about what causes the pain, and we still don't have any pain treatment that's really effective. And this is what led us to wanting really to investigate pain in a mouse model. And this is a different one from the one Mara so elegantly has developed. This is inducible. So it's a mouse model where one gives it a compound in the water, doxycycline, and this will induce the mutation in some bones in the skeleton. It is site-specific, so it will be induced in the cranium and in some of the long bones. This is a mouse model that was developed in the lab of Sylvia Goodkin and led by Sufeng Zhao. And in these mice, so we have when we induce the mouse that we call the FD mice, we see that they express the DNAS mutation. And if we give it to our control mice, they do not express the mutation. So we have FD mice that expresses the mutation in the sites that you see in the skeleton up here. And we have control mice which do not express the mutation. And this is very much a little bit of Chelsea's work that I'm going to show you. So here you can see that there is a progressive development of the FD lesion in male and in female and male mice. You notice the calcaneus is heel bone at baseline in the female, and the male is very sort of intact. And then after inducing the mutation, we see that it's sort of, they start having the lesions there, and eventually, as the next week, we'll see more bone affected. And we can see that from histology, and this is from Miguel's lab in Mexico. We can see that that we have a sort of classical FD lesions in the bone, which is also what they demonstrated in their original paper from the Goodkin lab in 2018, and we have the micro CT, the scanning of the bones, and the one that looks really intact are the the one that looks really intact are the control mice, and you can see the sort of the bones being affected by the lesions in the FD mice, both in female and male. So once we want to try to estimate whether these mice might have a pain phenotype, we rely on behavioral measures, and we use home cage activity and wheel running. And this is quite nice because the mice will be in their sort of environment that they know and not stressed by anything. And we just see how much they move around in their cage and how much they run in a wheel. We can, as Mara showed you, we can see how much sand they dig out of a tube, also their functioning and well-being, and we can see how much they hang in a grid. We have validated these assays, the burrowing and the grid hanging, with analgesia, so we can see if we give an analgesic, then that improves their functioning. So we think at least some of what the behavior that we see is related to pain in the mice. And I'm just going to show you one example, which is this one from home cage activity in the male mice, but we see a similar pattern in the female mice. So to the left, we see here the cage activity. And these you see, and the first ones you see here are the FD mice. And at baseline, they have an activity that was the sort of unit of 10. And then as the mutation is induced and the disease progresses, you see that the activity goes down. And this does not happen in the control mice. We also see that in the beginning, they run on the wheels, but this also sort of is diminished over time in the FD mice and not in the control mice. So, so this is the behavior. And now I want to talk a little bit about the studies that we have done in the mice, because when we look at what happens, we saw the beautiful nerve fibers in Mara's images. So we have the sensory neurons in the bone, and they can be activated by many things: injury, tumor, or in this case, by most likely a fibrous dysplasia lesion. At least that's the hypothesis. Then the signal will be transmitted up to the dorsal ganglions where the cell bodies of the neurons are, these afferent sensory neurons, and then onto the spinal cord, where the synapse was sort of neurons that one goes up and to the brain, where we perceive the signal and feel the pain. We can, if we think about what we saw in the patients, that some of the patients reported a more neuropathic-like pain that may be caused by nerve damage, that sort of inspired us to look first in the dorsal root ganglion, where the cell bodies are in the FD mice. And we measured the expression of a factor called ATF3, activating transcription factor 3. And you see here and this is also Chelsea's work. You see here the female. This is control female mice, and this is the FD mice. That's just a single cell lighting up with stain for this activation and this transcription factor. And this is actually also done in Miguel's lab. But if you look at the lower panel, we have the control male mice to the left, and to the right, we see that there's an increased signal for this transcription factor, and this can be quantified. Where to the right, you see here that that the male mice will have an increased level of ATF3 positive cell bodies, indicating that there is some kind of nerve damage happening. But one can also look in the spinal cord, and this was work done by Julie together with Chelsea, and she was actually, we were able to hire Julie from the grant that we got from the University of Pennsylvania Orphan Disease Center and the FDMAS Alliance for 6 months. And Julie looked at the level of the spinal cord, because we want to see if there were any central changes in the nervous system in response to the FD lesions. And by looking in the spinal cord, we can look both at the expression of neurotransmitters, and we can look at the cells, not the neurons, but the other cells, like what we call the glia cells, which are in the spinal cord. And so we sort of, Julie measured stained, as you can see below, many spinal cord sections from the control and from the FD mice for different neurotransmitters and for these glia cells. And she didn't find any changes, both for the neuropeptide expression and also for the glial cells. There didn't seem to be a difference in the expression between the FD mice and the control mice, indicating that there was no central changes that we could detect, and that whatever is happening to the nervous system is likely coming from the periphery, which has then made us go. So this was the sort of the first aim in the grant that we received. And the second aim was then to look at what the cells produce. So in this case, we took cells and isolated them from the mice and incubated them in culture dish and induced them with a doxycycline, so induced the mutation. And then we looked at this nerve growth factor. Nerve growth factor is among one of its functions that it sensitizes the neurons, the sensory neurons, and an antibody against this can be preventing the pain in other animal models of bone pain. And we found in these cells, we found that the intercellular cyclic AMP concentrations were increased as expected when we induced the cells. So the FD cells are the blue here to the left, and you can see when they are induced with doxycycline, they have an increased cyclic AMP, which we expect when in response to the mutation. And we don't see that in the control cells, and we don't see it when we don't give it doxycycline. We also saw that there was an increased level of the nerve growth factor produced from the cells. And this is corresponding to what Mara's group has found, that the paper she was referring to, where they found that there's an increased gene expression of nerve growth factor in the FD patients compared to healthy donors. So basically, NGF is overexpressed most likely both in the mice and in the humans. It sensitized the nociceptive neurons. It could be involved in the pain. There is an antibody that's been developed that blocks NGF in patients, and that's been showed quite good pain relief in patients with arthritis, but it had some side effects. So one cannot, it was never approved because they couldn't increase the doses to the fact that it was better than sort of normal pain medication. But it is possible to combine the NGF antibody in lower doses with another antibody. And so we have also measured other factors that the cells secrete, and we have the nerve growth factor here, but we also have tumor necrosis factor. And tumor necrosis factor is involved in inflammation, and thereby can sort of sensitize the neurons and cause some pain. And a company in the UK called AstraZeneca has an antibody that combines a block of both nerve growth factor and also tumor necrosis factor. So that inspired us to maybe we could try to treat the mice with both. So that is our long-term goal. What we have done for the aim 3 and 4 for the grant that we received was that we wanted to treat with a medication against the TNF alpha, the one that causes inflammation, and the NGF, the one that sensitizes the neurons, both by themselves and then trying to combine them. And then the second aim, then the fourth aim was, and then analyze the effect on the tissue. Of course, both on the pain behavior and also on the tissue. And we have we've gotten so far, because these are big experiments involving many, many mice, is that we have used an medication which is used to block TNF alpha. And Julie performed a big experiment. I cannot show you the results. We haven't got to the publication level yet. But she measured, she treated the mice 3 times, and she did behavior measurements. We also had Miguel in Mexico and his team analyzing both for the nerve fibers, for the immune cells and osteoclasts, and also for the effect on the structure of the bone. So this is work that's going to be continued. And the next step is that we have many things that we want to do with the FD mice, but with the focus of this treatment, we want to test the efficacy of medications against both nerve growth factor and combining this with the medication against TNF alpha in the mouse model of FD that we're using. So I want to again thank the research grant that we received, because it was able to provide bridge funding for us. It was able to make us perform the experiments that I have talked about today. And it also enabled us now that we actually have EU funding for a PhD project led by Yingyu Tang over the next 3 years, where we are going to continue our studies with pain in this model of FD, and also expanding into human tissues. So thank you.
I'm struggling to unmute myself and turn my camera. Thank you, Emery. That was really nice. Again, a really crystal clear presentation. You've made amazing progress. I'm really impressed, and obviously the ability to translate this work to the clinic is looking very promising. Wonderful presentation. Thank you. Let's see if there's any questions in the chat from Cindi. Cindi, are you still here? You want to just ask your
Yes,
question?
I am. Yeah, I didn't know if I'm explaining my question well, so that's why I tried to put it in writing, if it makes any sense to you scientists there. So as we know, there are patients who have extensive disease, do not experience pain. And then we have ones who have localized 1 or 2 spots or whatever and have a lot of pain. And just wondering if there's any research being done to try to capitalize on that as figuring out why the pain is caused in some and not other FD patients.
That is the question. I mean, this is, and it's not just for FD, it's also for osteoarthritis. You can have people with osteoarthritis where you can basically, it looks like the bones are just sort of grinding against each other and they have no pain. And you can have some where the joint looks quite nice and they have pain. We have it in cancer. You can have patients with bone metastasis where the cancer is spread to the bones, where there's actually no pain, and then you can have other with just one single. We do not know the answer for that. But I mean, if I could ever solve a question in my scientific life, that would be the one I would like to solve. But we do not know. We're looking into it in others, also other bone painful bone diseases, but we don't have an answer. And I think Mara would like to study that in her mouse model, right?
Mike, can I make a comment?
Yes, Michael White, please go ahead.
Yes. So whether you're dealing with a mouse model for fibrous dysplasia or patients themselves, I wonder if the field needs to think about the skeleton a little bit beyond just the bone to the entheses. Your mouse model looked like it had a terrible Achilles enthesopathy there. And as you continue to study, you might want to explore cells that come out of the enthesis itself, where the tendon is transforming into bone-like material and then finally attaching to the bone itself.
That's a very good suggestion. I'm making notes. Definitely.
Could we also collaborate with any other groups of diseases, cancers, things like that, to facilitate some of this research through the Alliance, but not being such a rare disease where we can't get funding? If there's any suggestions anyone has here, we welcome them.
I mean, I'm a coordinator now for the third time for a European network of researchers where we've trained PhD students within bone pain. And it was always sort of the thought that we have people working on osteoarthritis, I work also on cancer in the bone and in my group and rheumatoid arthritis. And I always think that what we find is going to cross-fertilize across the different painful bone disorders. So we're definitely thinking that way.
Thank you. Thank you very much.
Fernando.
Anne-Marie, that was beautiful. Thanks for the presentation. I wonder, when you said that the NGF was increased in cells from the mice after treatment, is that right?
But
So
when we induce, I mean, and they've also seen that in Mike Collins' lab, and Louis has seen that, and also in I think from patients' cells, they've seen the same. And we also know it's when you induce the mutation in the mice. Well, you reduce the mutation in the cells, then we see that they express more NGF. So basically, the bone marrow stroma cells that we isolate from the mice induce the mutation in with the doxycycline, and then we measure what they have an increase in the NGF.
Yeah, perfect. So one question was that the cell type, and then what I didn't understand well, doesn't sound intuitive to me that the nerve growth factor, nerve growth factor is inducing nerve damage. Is that what you suggested?
No, it's sensitized. It does actually several things. It induces nerve outgrowth. So we actually see, and this is what Mara has been looking as well, both in the patients and in the mice. in other mouse models and also some human samples, for instance, for arthritis or for cancer, we see that nerve growth factor drives what we sort of call like a nerve sprouting in the bones, specifically in the periosteum, in this outer sheath of the bone. But it's also well known in non-bone disorders to cause sensitization of the nerve. So you can, if they basically sensitize the peripheral neurons by, changing the receptors and changing the trafficking and how the level of reaction. So by blocking nerve growth factor, you can block the pain.
But without causing a neuropathy or something like that, something that
Yes,
I would
yes.
imagine that neurons need this factor. That's why.
Yes, and that's a big question. That's the issue, because there's an antibody that was taken all the way to FDA approving, and the patients that get high doses, they get what is called a rapid progressing osteoarthritis. So they had to lower the dose in the clinical trials, and in that way, the dose was not higher. That the efficacy was not better than it was sort of estimated than you would get with no other analgesic out there. So it wasn't approved. So there is, in high doses, there is a risk for this rapid progressing osteoarthritis, and that is why the scheme that AstraZeneca has been promoting, which is online in their pipeline, I don't know if they're still continuing with it, is that if you can go lower dose with the anti-NGF antibody and combining it with an anti-TNF alpha, then you get a good pain relief, but without the risk of the side effects.
Great.
That's what we want to try in the mice.
Yeah.
Great. What about the concept of delivering this antibody to the affected tissue or local administration to bone tissue, maybe even taking the approach that Dr. White took with alphatase alpha, the way they delivered the alkaline phosphatase to bone tissue. So combining the antibody with something that will make it home to the bone, so you can get high levels at affected tissue, but not get the systemic effects.
I think the issue with the anti-NGF is actually that the effect is that the side effect is also coming from the bones.
What do you mean the side effect is coming?
I mean, but in the clinical study, they had more patients who had to have joint replacements in the treatment group than the non-treatment group. So the side effect of the antibody treatment is related to the bones.
Yeah, I mean, way I interpreted those studies is that those patients with arthritis were having less pain now, so they did more.
That's a hypothesis. That's a hypothesis.
No,
yeah.
But that's a hypothesis that could be tested in animals without worry of
absolutely. But anyway, to home something to the bone is always a beautiful idea. I would love to do it.
Yeah. I had another sort of technical question. So when you showed that there was, I think it was in the dorsal root ganglia that you showed there was diminished expression of those ATF3 expressing cells.
No, increased.
When you see that, would that cause more or less pain?
That would, like in other models, like the models of neuropathic pain that you can also have in mice, you expect this to be causing more pain.
Okay. All right, so that makes sense. It doesn't look like there's any other questions in the chat. And we have about 4 minutes left. And, I did have a question related to Cindy's question, which is the sort of insightful questions that you always get from parents of kids or the patients themselves. And, probably because the mouse strains, do you think that the patients who have a lot of bone disease and no pain, that they've just sort of burned out their pain, or do you think there's something genetically different about them?
The honest answer, I have no clue.
And towards that end, I mean, because with these inbred strains of mice, you wouldn't see those sort of outbred genetic differences. But do you ever see mice that are sort of outliers and they're not behaving like they should be, and maybe those should be the ones to go after?
I mean, if you look at Mara's study, at what they have been doing, where they have, they have this mouse behavior deficits and they have no correlation with the disease burden, right? So maybe there's something to go in those mice.
And Mike, at the NIH, do you have such patients that don't have the pain? And if you did one of your fluorine PET scans, are they cold as opposed to hot and patients with fibrous dysplasia and pain?
No, they have tons of bone activity in the absence of pain. We saw the same thing in humans. We saw no correlation between disease burden and degree of pain. It was quite surprising. But
so is somebody tackling nociplastic?
Yeah, so I think that a couple groups, so Kassim Javed is interested in that, and Jamin at Boston Children's is interested in that. They're addressing that. I don't know this area well, but my understanding is that those sorts of pains respond well to different classes of medications. And I think that Jamin is looking at that. He's studying patients with that sort of pain. They're identifying those patients and they're treating those patients and doing brain scans, et cetera, before and after certain treatments. So Jamin, I think, was also a recipient of $1 million bike ride grant, and I think now also has a larger grant. So again, MDBR has really fostered progress in this area.
Thank you.
Well, we're almost right at the hour. So Tova and/or Adrian, would you like to close this meeting?
Yeah, thank you so much, Mara. Thank you so much, Anne-Marie. Mike, thank you for leading this discussion. I think, hopefully folks have had a chance to see that we do have more exciting research news coming soon. And we hope that this group that's especially committed to FDMAS research will stay with us through all that journey. In particular, within the next month, we expect funding announcements from the Department of Defense, PRMRP, the Peer Reviewed Medical Research Program, and that funding, which we've already seen previous Team FDMAS awardees go on to receive, is all made possible because of the alliance, the advocates that the FDMAS Alliance has sent to speak about the importance of this federal medical research funding, as well as, of course, the NIH funding. If you do decide to apply for the Department of Defense funding, we hope that you'll reach out to us. We have resources to support your work, and please let Adrian know if you're interested in that. Adrian, I don't know if there's anything else you want to add. But yeah, I think, when you're supporting FDMAS Alliance and our research programs, it's not just these seed grants, it is that federal research funding, the advocacy work that ensures that there are bigger places for this research to go on and continue, and hopefully make it eventually to the community at large. I'm so grateful. I'm again putting the launch of our 2020. Oh no, I'm so sorry. That is actually the 2025. So don't ignore that one. Let me put the 2025. It'll all go to the same place, but this has this year's event details on it. And yeah, we're so grateful and can't wait to see you out there and at next year's research update.
Thank you, Tova and Adrian, for all that you do. And I'm sorry for your poor doggy, Adrian, with that terrible collar on.
Yeah, he's got bone issues. So,
All right.
Be well, everybody. Thank you.
Thank you. Bye-bye.
Everyone.